Okay, thanks everyone for being here. My name's Taylor. I'm a postdoc in Ben Leonard's lab, just upstairs here. And I'm going to be talking about what I think is a pretty interesting paradox where we can actually rescue the expression of mutant GPCRs with antagonists. Okay, so just to speak a bit about, I guess, why we're all here, which is the fact that genetic disease is quite complicated, and this figure is out of date, but the point is that there's many different disease genes falling into many different classes, and it feels quite daunting to look at this and imagine how we can make progress toward treating all these diseases. But as Creston pointed out very nicely this morning, even though there's a huge diversity of disease genes, it actually looks like there's one predominant disease mechanism, which is loss of stability. So we're sort of converging between computational and experimental methods on an estimate of about half of all pathogenic variants being pathogenic just because they lose stability. Okay, so now this pie chart looks a bit different, and it maybe gives us a little more hope that by looking at this one really predominant phenotype, we may be able to start making some progress, okay? So I'm really interested in GPCRs. These are G protein coupled receptors. So these are transmembrane proteins that sense molecules in the extracellular environment and transmit that information into the cell. In the wild type case, these things are synthesized and folded in the ER and then trafficked to the plasma membrane. But we know that oftentimes when GPCRs are mutated, they don't fold quite properly, and the trafficking is disrupted, and they never actually make it to the membrane, okay? But one interesting thing that's been known actually for decades is that if you treat a cell carrying a mutant receptor with an antagonist that can cross the plasma membrane, it can actually bind to the receptor in the ER, stabilize a conformation, and allow the receptor to actually make it to the membrane. And the way we understand this is quite simple. So basically, GPCRs, like many proteins, are quite dynamic. In the unbound state, they're shifting conformations between various different conformations. And when we can bind a high-affinity antagonist, we can shift this equilibrium into a low free energy, highly stable, inactive conformation, and this allows trafficking to the membrane. So this is, as I mentioned, this has been known actually for a really long time and has been demonstrated for many receptors in many contexts. So here I'm showing the rescue of this rhodopsin variant with this antagonist in human cells. But this also works in animals. So mutations in this GPCR cause this disease, basically. And if you treat with this antagonist stabilizer, you can actually reverse the phenotype in a mouse. Okay, and it's not only mice, this has also been shown in humans. So mutations to the vasopressin 2 receptor cause a disease of water homeostasis. And the details here are not important, but this is a clinical trial from 2006 showing that an antagonist stabilizer of this receptor can actually improve water homeostasis phenotype in humans. So unfortunately, this was discontinued due to off-target side effects, but the principle was shown that this can actually work in humans. So the question is, why aren't we doing this more frequently for more targets across different diseases? And the answer is that until very recently, we didn't know the answer to either of these questions, which are sort of key before we want to embark on developing a clinical therapeutic. So first, we want to know for any given disease gene, how many of them are actually pathogenic because they lose stability. And then out of that population, how many can we, in principle, rescue with a stabilizer? So you probably see where this is going since we're at the Mutational Scanning Symposium. We think that mutational scanning is the way to answer these questions, okay? So we set up a platform to look at the vasopressin 2 receptor. So this is what I just mentioned, has been successfully drugged in a clinical trial. Mutations here cause a disease of water homeostasis, and there's really no treatment, no direct treatment for this disease. So I wanted to see if we could make some progress here. I'll just go through the pipeline quickly. So as Max mentioned, we use this really nice mutagenesis technique called Sunny mutagenesis. We put the receptor in a construct with an HA tag and a unique DNA barcode. We put these into Kenny Matrix landing pad cells and then do a traditional sort-seq experiment. And this works pretty well. We get nice data, which makes sense according to what we know about GPCRs, which is, for example, that the transmembrane helices are expected to be sensitive to mutation, and indeed they are. And of course, the controls look basically how we expect them to look. Okay, so taking a closer look at the human variants, we can split these into different categories. So basically, 3 categories which we presume to be benign, and 3 which we presume to be pathogenic based on NOMAD, ClinVar, or human gene mutation database. You can see the distributions are obviously different. When we categorize the variants, we can also see that for the benign categories, most of the variants are well expressed, like we would expect. But in the pathogenic classes, we see sort of exactly aligning with our prediction, about half of them are poorly expressed. This is sort of just an additional way to look at this. We can make ROC curves trying to discriminate pathogenic from benign alleles. And the VEPs, as I'm sure all of us are well aware, they do actually really well. They do better than the surface expression alone, but the surface expression measurements do better than the stability estimates. And this probably makes sense because these, in general, are not trained on membrane proteins, and probably membrane proteins are a bit more difficult to predict. But anyway, we can be fairly confident that at least for this gene, we can answer this question, which is what fraction of pathogenic variants are loss of stability? And it looks like, consistent with our expectation, it's about half. Okay. So now I wanted to see if we can see this rescue phenotype, and I was interested in this drug, tolvaptan, because it's actually an approved drug, and it's used to treat polycystic kidney disease, which is a disorder where the vasopressin signaling is actually elevated. And it's been shown that if you treat cells expressing wild type or variant vasopressin receptor constructs, and treat with tolvaptan, you can rescue, for example, this L44P variant to the membrane. In some cases, it works well. In some cases, it works less well. So basically, this led the authors to speculate, well, it's difficult to predict. We're not really sure how generic this rescue is. But of course, with DMS, we can just go ahead and answer the question for all the possible variants. So we did the exact same experiment, but this time we treated the cells with this tolvaptan, and just even at the facts level, when we're comparing the population of cells treated with tolvaptan with the control, we can already see there's a substantial shift from low expressing to high expressing variants. Okay. And then the really exciting result is that when we do the sequencing and the quantification, and we compare the expression level of all the variants in the presence of tolvaptan compared to the control, we see that there's a very large, widespread shift in the expression level of these variants. So this is looking at all possible missense variants. This is looking at just the variants that are known to cause this water homeostasis disease. And again, we can see that we're rescuing a vast majority of these variants. If we categorize them as we did before, in the control condition, again, we see about half of the variants, of the disease variants are poorly expressed. In the tolvaptan condition, we move about 90% of these to at least this moderately expressed category. Okay, so we think from this, we can actually answer the second question, which is what fraction can be, in principle, rescued by a stabilizer, and we think it's a huge percentage, like about 90%. Okay. You may have noticed that, of course, not every single variant is rescued, and we were quite curious to understand basically what these variants are. So we devised an approach where we just fit a low-S regression to the data that's shown in black here. And then basically we looked for positions, for example, this S171, where variants are consistently rescued less than you'd expect, right? So the model, the black line is the expectation, and all of these variants at this position are in orange, and they're basically not behaving as we expected. Okay, and this is just a volcano plot showing the different positions that we identify. And the cool and kind of nice thing is that the sites that are not rescued seem to make perfect sense, okay? So in the N-terminus of the receptor, there are two well-known glycosylation sites, so O and N glycosylation. And here I'm showing sites that are not rescued in red according to the strength of the p-value. And we can see that, of course, the glycosylation sites are not rescued by the small molecule because a small molecule cannot compensate for a lack of post-translational modification. The other place where we see an enrichment of these non-rescued sites is actually just in the binding site of the tolvaptan molecule. So again, this is sort of a trivial explanation, means like there's nothing complicated going on here. It's just small molecules cannot compensate for post-translational modifications, and a small molecule cannot rescue a receptor if it can't bind to the receptor. Okay, so we think this is quite good news. We think that hopefully this will be true for other receptors and other proteins. And we think the fact that all of this is so simple supports that notion. Yeah, this is pre-printed and accepted for publication, so if you're curious to take a closer look, I encourage you to do that. This sort of raises a handful of questions. Some of these we've made progress on, some of them we haven't. So first, you might just ask, is this a one-off phenomenon? And at least for the vasopressin receptor, and at least for one other stabilizer, I can say that it also works. Another question that's of obvious importance is like, sure, we got the receptors to the membrane, but can they actually function once they get there? We are working on this, and I have something to show you related to this. And then finally, we would just like to know how general this phenomenon is among other GPCRs and, of course, other classes of proteins. Okay. So back to this point, like, are the rescued variants functional? So in order to get at this, we needed to develop a deep mutational scanning style approach for measuring GPCR signaling. This is all not important. The important part is like we can express GPCRs in cells, stimulate with drug, and read out their signaling through barcoded RNA. So this is important because it means that we can really scale these experiments. This is just an example of a signaling data set of the vasopressin receptor in response to its endogenous ligand signaling through one of the canonical pathways. You can see that the data overall is quite nice, but signaling is not a phenotype that's well captured by one measurement, so we were really interested to see if we could push this to proper like dose response analysis to get a handle on different pharmacological parameters, and we were able to do this. So now we have here on the x-axis positions in the receptor, y-axis all the mutations, but now at each site we have 12-point dose response curves, right? So we can start thinking about efficacy, potency, et cetera, of all of these variants. And of course, because it's deep mutational scanning, it's highly scalable. So here I'm showing just a few hundred, but we have at this point about 5,000 of these high-quality dose response curves. So this preprint is also coming very soon. If you're at all interested in GPCRs, I encourage you to take a look. With that, I would just like to thank the Lenner Lab Empire, and thank you all for being here, and I'm happy to take any questions.